Good afternoon. My name is Khan Kutlu. I'm a student at Miami-Dade College, Wolfson. I worked alongside Ian Ballard, Blas Vargas, and Jason Mendoza on our project on enzymes. This is for BSC 2010L. Um, we were mentored by and sponsored by Professor Juan Morata. Um, this is our presentation on enzymes. We'll be explaining our experiment here. We're going to analyze the data and break down the significance of the biological findings. So, without further ado, we're going to move on to the abstract and the hypothesis.
Hello, my name is Jason Mendoza. I'm a student here at Miami-Dade College Wilson campus, and I was talking about at the abstract where enzymes are first of all are proteins that speed up chemical reactions and usually work with only certain substances. In this experiment, we tested how lactase works by using lactose and sucrose solutions to see if lactase breaks down both sugars or just one. Five wells were set up with different combinations of lactose, sucrose, and lactase, distilled water, and a sample with glucose as a positive control. After 5 minutes, glucose test strips were used to check whether glucose was produced in each well and how much. Breaking down lactose with lactase makes glucose. Finding glucose showed that the reaction worked. The experiment made it easy to see the differences in glucose levels between the wells to find out if lactase works only on lactose or also on sucrose. The results show that how you can test what an enzyme works on using simple tests and a careful setup.
Well, my name is Blas Vargas. I'm a student from MDC College in Wilson campus. Now, today I'm going to be talking about the introduction and methodology. In the introduction, in this experiment, we focus on enzymes, specifically how they work and how specific they are. Well, enzymes are basically biological catalysts, which means they speed up chemical reactions by lowering the activation energy. They help reactions happen faster and more efficiently inside living organisms. The enzyme we look at was lactose. Lactose's job is to break down lactose into smaller sugars. It will have the glucose and the galactose. It can only do this because its active site is shaped perfectly for lactose, kind of like a lock and key situation. So the main goal of the experiment was to test that specificity by comparing how lactose reacts with lactose versus how it reacts with sucrose. Now, for the methodology, so for the experiment, we create a control lab setup to basically test the glucose, doing a results and model, and what we'll expect to see when testing how lactose works. We use five different conditions. The first one is we use saccharose with distilled water. The second one was lactose with distilled water. The third one was sucrose with lactose. The fourth one was lactose with lactase. And the last one, we did glucose as a positive control. Now, for each of these, we made three pretend trials just to show what normal operation in glucose test strip readings might look like. All the glucose levels were written as realistic milligrams values. We kept the readings really low or close to zero in the condition where glucose shouldn't appear. In the lactose with lactase condition, the readings were higher since that's where the enzyme will actually break down the lactose in glucose. And for the glucose and the positive control, the numbers were very high. Now, after that, we averaged the three measurements for each condition to show the typical glucose level. And overall, this fake database helped us to understand what we should expect in a real lab and made it easier to see how lactose works and what substrate it acts and in a controlled way.
Hi, I'm Ian, and I'm also a student at Miami-Dade College Wolfson campus. I will be reading the conclusion or expected outcomes of our experiment. The results of this experiment showed that lactase only works with certain substances, breaking down lactose but not sucrose. This demonstrates enzyme specificity, Wells with lactose and lactase, well D, clearly had glucose, which means that the lactose was successfully split into glucose and galactose. This confirms the reaction occurred as expected. In comparison, wells containing sucrose and lactase, well C, showed little or no glucose, indicating that lactase does not break down sucrose. The enzyme simply cannot bind to sucrose structure. The wells with only lactose or only sucrose, wells A and B, also do not produce glucose, proving that the enzyme is needed to break down these sugars. Without lactase present, no glucose forms. As we can see in graph number 1, the glucose was created in higher amounts with the lactase and lactose, and the positive control had much more glucose than that. And then in the AI results, similar results were found, but with lower numbers, which shows that AI can give us an accurate trend into how the experiment should work, but with specific numbers, it may be slightly inaccurate.
This is a very big experiment for us. We used a couple references, notably was Microsoft Copilot, ChatGPT, Khan Academy, and StudyMonkey. As for acknowledgements, we would like to point out that this research is funded by Coalesce. This is a program of the STEM Legacy Grant Project at Miami-Dade College, Wolfson. And additionally, this work was inspired by the enzyme specificity and experimental outline in the HOL Science Interactive Platform, and was also supported by the guidance from the Miami-Dade College Laboratory Protocols and the Department of Natural Sciences. We would like to give a special thank you to Professor Juan Morata for his support and thank the STEM Legacy Program for promoting hands-on learning experiences here at Wolfson. Thank you so much for listening.